Okay, students, am I audible? Is okay. So, good evening, everyone. So, today we are going to study one of the most important topic, and that is also comparatively important for your board level as well, right? And the topic which we are going to discuss today is glycolysis. Before starting with glycolysis, I'll first let you know like what are all the things you need to understand. Okay, see, when it comes to NEET point of view, or as I said, like this is an important topic for both NEET as well as for your boards, right? In case of boards, it is going to be your 5 mark question. This is going to be your 5 mark question. Whichever the board you are from, whether it is you are from CBSE or state board or from IGCSE, whatever it is, this topic is very common. Okay, glycolysis. So first I'll break this word. The word glyco means, the word glyco means we are talking about glucose, right? And the word lysis means breakdown, right? So with the name itself, we understand what we are going to do with this glycolysis. We are going to break glucose. Simple, right? Second question, why glucose has to get breakdown? First thing, we know that plants are the producers. Plants are producers, right? And these plants produce food in a form of, consider glucose. They produce the food in a form of glucose, and they'll store in a form of sucrose. Got it? And to produce this glucose, plants undergo a process called as photosynthesis. To produce this glucose, they undergo a process called as photosynthesis. And to produce one molecule of glucose, they spend more amount of ATP and NADPH. By spending more amount of ATP and NADPH, they produce glucose. For example, if I say to produce one molecule of glucose, we know that cycle is called Calvin cycle, and this Calvin cycle, which takes place for 6 times. It takes place for 6 times, right? And for every single time, for one carbon dioxide, they'll be utilizing, they'll spend, okay, they'll spend 3 ATP and 1 NADPH. Okay, so this is for one carbon dioxide. So, and we know that glucose is 6 carbon molecule, right? So in that case, if we multiply this for 6 times, okay, so nearly 18 ATP is spent and 16 2 NADPH, I'm sorry, this is, yeah, they spend 2 NADPH for one carbon dioxide, and for 6 times it will be 12. It will be 12 NADPH will be spent. So by spending this much of energy, they produce one single molecule of glucose. By spending this much of energy, they produce one single molecule of glucose. Okay, and so from this glucose, we need to get more energy. We are going to break down this glucose, and we are going to get energy from this glucose. So this is what this glycolysis is all about. I hope this one is clear, right? So next thing, this glycolysis is also known as EMP pathway. Means this glycolysis breakdown process been explained by 3 scientists, Emden, Meyerhoff, and Parnas. So these are all the 3 scientists who explained this glycolysis process. So this is also called as EMP pathway, right? And next thing, we need to understand this. Glycolysis is universal. What does it mean? This glycolysis happens in all organisms, right? If I talk about bacteria, yes, they undergo glycolysis. If I talk about a plant, yes, they undergo glycolysis. If I talk about an animal, yes, they do also undergo glycolysis, right? So, any organism you listed, all organisms will undergo this glycolysis process. So, I call it as universal. Okay, and one more thing, this glycolysis process is oxygen independent. It is partially oxygen independent process. Okay, so if I talk about this glycolysis, not the whole glycolysis will require oxygen. A limited amount of glycolysis pathway requires oxygen. For example, this glycolysis I'll break down into. This glycolysis process will be break down into two steps. One is preparatory phase. Another one is payoff phase. So the whole glycolysis will be break down into two steps. In preparatory phase, we are preparing the glucose to get breakdown. So in this case, we are going to spend energy. Whereas in payoff phase, we are going to start gaining energy. Okay, when we are spending energy, we know that ATP is the energy currency, or the form of energy is ATP, right? Okay, so here we will spend ATP, and here while we are gaining, we will gain in two forms. One will get at a substrate level, or another one is a oxidative level. One is substrate level, another one is oxidative level. Substrate level is nothing but when the cycle during the process itself. Okay, before completing the process, you will get the energy. Okay, consider for example a process starts, and when the process happens, by the time itself you will get energy currency, okay, which can be utilized, okay, which can be used. And in another case, oxidative means you will get your energy, but you cannot use at the moment. You need to break it further. Okay, for example, consider how I say to my students are, like, consider you are going to have cash in two form. Okay, one is handy cash, okay, a normal regular note. Okay, consider 100 rupees or 200 rupees note. Okay, and another one thing, I'll give a credit card or debit card to you. Okay, and that debit card also has some amount of money. Okay, but if I say like which one you can utilize right moment, okay, the thing is you will say the amount which you have handy. Okay, and even if you want to spend that amount which is in the debit card, you need some ATM or any POS machine to utilize. Similarly, ATP is the substrate level energy which will be produced during the process, and oxidative level energy, which is NADH2, this will be produced during the process, but you cannot utilize this at this moment. You need to oxidate it. That means each one NADH2 is equal to 3 ATP. That means if I break this one NADH2, I'll get 3 ATP, but I will not get at this moment because I'm doing this process. Once I complete this process, then I need to take it to another one process called electron transport system, and there I'll get this into 3 ATP. I hope you understand, right? So make it very clear, glycolysis is of two steps. One is preparatory phase, another one is payoff phase. In preparatory phase, you will be spending energy. You are preparing the glucose for breakdown, and payoff phase you will start gaining energy. While you are spending, you will spend in a form of ATP, and while the time of gaining, you will start gaining in two form. One is in a direct form, that is ATP. Another one is in an oxidative form, which is NADH2. Got it? So this is all the basics which you need to know before understanding glycolysis, and I hope this is clear so far. Yeah. Getting into the core topic, glycolysis. And here first I'm going to start talking about preparatory phase. Okay, and one more thing I wanted to add here, Glycolysis is a enzyme catalyzed process. Enzyme catalytic process. What does this mean? Every single step, okay, when a glucose is getting breakdown, under every step one enzyme will be included or one enzyme will be involved during this process. You may ask me why. The thing is, this glucose, consider I have one molecule of glucose. I have one molecule of glucose. Okay, if I break this glucose, so what I'll get some amount of energy. But what I'm going to do is I'm going to break this glucose in a small steps, and so I will gain more energy. So every single step will have another form of enzyme which will help us to get more amount of energy. So remember, glycolysis or glucose breakdown process is an enzyme catalytic process. I hope this one is clear. So you may get a question like this: why enzymes are involved in the glycolysis process? Because to increase the energy efficiency. Okay, fine. So coming to the preparatory phase. Okay, so first we know that which substrate we are going to use here. The substrate is nothing but your glucose. What is substrate? Substrate means the substance from where we are going to undergo this process. That is called substrate. Okay, so here we are going to use this glucose for this breakdown process, and this glucose, how many carbon compound? It is 6 carbon compound. How many molecule we have? We have 1 molecule of glucose. Okay, so consider now this glucose is going to get converted into glucose 6 phosphate. What does it mean? Why a glucose should become glucose 6 phosphate? Okay, firstly, understand that in the whole universe we have n amount of glucose. Okay, and not all the glucose are readily available to produce energy. Okay, or else you can also say in such a way, I need to occupy some amount of, I need to accumulate some amount of glucose for my energy process. Okay, so in that case, what I'll do first, I'll spend some amount of ATP and I'll make the glucose in a form of glucose 6 phosphate. Got it? So since I have added 1 phosphate, okay, you can see ATP is 3 phosphate and ADP is 2 phosphate. So where is that 1 phosphate? That 1 phosphate is this. Got it? So I got glucose got converted into glucose 6 phosphate. Remember, this is a first regulatory step. This is a first regulatory step. What does it mean? Regulatory step means these steps are going to control this process. Okay. Likewise, we'll be having 3 steps. If I say glycolysis is a 10-step process, under this 10-step process, we have 3 steps which are considered as a regulatory steps. That means these steps are going to regulate the process. If they want to stop, they will stop from that. If they want to start, they will start from that. Okay, and regulatory steps are irreversible. Remember, regulatory steps are irreversible. So how many irreversible steps are there in glycolysis? It is 3 steps. So which is the one? The first step is a regulatory step. That means it is irreversible. What is that step? Glucose gets converted into glucose 6 phosphate. Clear, right? Next thing, this glucose, and for this process, to undergo this process, we need an enzyme called as hexokinase. We need an enzyme called hexokinase. Okay, as I already stated, every single step requires an enzyme. Okay, so hexo, in sense, we know hexo is 6. Okay, that means I'm going to break a 6 carbon molecule. Okay, and kinase means I'm going to break it. Okay, so in the time of breakdown, or consider whenever I do kinase, so at the time I'll be either adding ATP or I'll be gaining ATP. I mean, like, I'll utilize ATP or I'll gain ATP. Got it? So first thing, it is hexokinase. So glucose 6 phosphate it get produced. Okay, and this glucose 6 phosphate is getting converted into fructose 6 phosphate. This gets converted into fructose 6 phosphate. What is this? glucose and fructose, right? And both this sugars are 6 carbon compound. This is also 6 carbon, this is also 6 carbon compound. That means what happens here, this is nothing but the enzyme which is called isomerase, which involved in converting the glucose into fructose, because actually we are talking about preparatory phase. Okay, I mean I'm going to prepare the glucose to get involved in this breakdown process. So I cannot break the glucose as such. Instead, I'm going to convert it into fructose and then I'll start breaking it. So I'll convert the glucose 6 phosphate into fructose 6 phosphate, right? And now the next thing is fructose 6 phosphate gets converted into fructose 1 6 bisphosphate. Fructose 1 6 bisphosphate. What does it mean? Bisphosphate. That means we are going to add, we have 2 phosphate. Okay, so you can see that with glucose we got 1 phosphate and now we have another 1 phosphate. What does it mean? In this step we have added another 1 ATP. Okay, and we got ADP back. Got it? Fine. And what is this 1 and 6? That means in the first carbon, already you know that in the 6th carbon position, 1 ATP has been added. Okay, I mean like 1 phosphate is added. Now in the first carbon position, another 1 phosphate is getting added. How to make it easy? Let's say for example, this is 1 glucose. Okay, this is 1 glucose, right? And this is the carbon position 1 2 3 4 5 and 6. Okay, this is the carbon position. So when you see in this glucose, I'm going to add a phosphate. In which position? In 6th position. So here you can see, here I have phosphate. I have added the phosphate in the 6th position, right? Now I can see fructose 1 6 biphosphate. Okay, so what happens in the 6th, already in the 6th position I do have 1 phosphate, and now in the 5th position, first position also I'm going to add another phosphate. So this is called 1 6 bisphosphate. Actually this is a glucose structure. We need to consider this in a fructose structure. Hope you understand this, right? So remember, in this step, this is again a regulatory step. Okay, so I'll do one small change. This step is going to be reversible. Which step? Glucose 6 phosphate gets converted into fructose 6 phosphate is a reversible process. If it is reversible process, we don't consider that as a regulatory process. Okay, fine. And the second regulatory process we got here, second regulatory step is this. This is our second regulatory step. What is that? Fructose 6 phosphate gets converted into fructose 1 6 bisphosphate. Okay, so this is a second regulatory step, and remember this is a reducing step as well. Okay, RDS step. Okay, fine. So coming back to this, fructose 1 6 biphosphate gets converted now into, it gets breakdown into glyceraldehyde 3 phosphate or dihydroxy acetone phosphate. Okay, this fructose 1 6 biphosphate gets divided into glyceraldehyde 3 phosphate and dihydroxy acetone phosphate. Okay, so here you can see glucose is 6 carbon, glucose 6 phosphate is 6 carbon, fructose 6 phosphate is 6 carbon, fructose 1 6 phosphate is 6 carbon compound. But now what happened? Each are going to be 3 carbon compound. That means what I did, one single molecule I break into 2 different molecules. But the thing is, both the molecules are 3 carbon, which is again an isomer. Hope this one is clear, right? So what does it mean? Here, this glyceraldehyde 3 phosphate and dihydroxyacetone phosphate are both an isomers. Okay, what is isomer? Isomer is structurally different, or you can say different structure, same function. Okay, And here why it is one is glyceraldehyde, another one is dihydroxyacetone phosphate, because we know glucose usually has two functional groups. Glucose usually have two functional groups. What are all the two functional groups? One is aldehyde group, one is aldehyde group, another one is ketone group. So the glucose, which is in a preparatory phase, has prepared itself into its two functional group. So what is the complete motto of this preparatory phase? This preparatory phase complete motto is to get convert the glucose into a preparatory form where it is going to get breakdown. Okay, so if you ask me, sir, directly can I break the glucose? Yes, you can directly break the glucose, and directly you will start receiving energy, but the amount of energy which you will receive is very less. Okay, but when you go under this preparatory phase and you start doing that in a small steps, you will start receiving more number of energy. Okay, so with this your preparatory phase is getting over. With this your preparatory phase gets over. So how many step process it is? How many step process it is? You can see this is your first step, this is your first step. Okay, or you can say like this. Yeah, so this is step 1, this is step 2, this is step 3, and this is step 4. Got it? Actually this will be step 4. 1 minute. Yeah, so this will be step 4, and then the isomerase is the 5th step. So total how many steps are included in the preparatory phase? It is 5-step process. So how many steps are preparatory phase? It is a 5-step process. Okay, how many steps are in the total glycolysis pathway? 10-step process. So you understand total how many phases we do have? We have 2 phases. One is preparatory phase, another one is payoff phase. So remember, preparatory phase is 5-step, preparatory phase is 5-step, and payoff phase is also 5-step process. So totally it is 10-step process. Hope this is clear. Yeah, fine. So with this we are getting into the like preparatory phase, and you can see one more thing. Here we have spent energy. That means here we have spent, utilized, we have taken from our pocket and we utilized in the process. Okay, how many ATP we have utilized? We have spent 2 ATP. Okay, but what is the purpose of glycolysis? The glycolysis is to gain ATP. But remember one thing, in this preparatory phase we are not going to gain anything, we are going to spend. Okay, whereas in payoff phase we are going to gain. Okay, how much we have spent as of now? We have spent 2 ATP. So since we have spent, we'll consider that into minus. So how many ATP is spent? 2 ATP is spent. Okay, in which are all the step? In the first step and in the third step. Which are all the step? First step and third step. Remember, both these steps are regulatory steps. Both these steps are regulatory steps. Hope this is clear. Right? Now getting into payoff phase. Okay, now getting into payoff phase. I hope this is clear for you all. And now let's get into payoff phase. Okay, and this payoff phase starts with glyceraldehyde 3-phosphate. Why not dihydroxyacetone phosphate? You can go with that also, but anyway both are same, functionally both are same, right? So we'll take this as the substrate now, and this glyceraldehyde 3-phosphate is getting now converted into 1 minute. Excuse me. Okay. So in this phase, we are going to in this phase, this glycerol 3 phosphate is going to get converted into 1,3 biphospho glycerate. Is going to get converted into 1,3 biphospoglycerate. So what happens in this step? Remember, this is the sixth step. This is the sixth step. Okay. In this sixth step, what happened here? We are going to use an enzyme called as glycerol 3 phosphate dehydrogenase. The enzyme which we are going to here use it glycerol 3 phosphate dehydrogenase, and because of the term dehydrogenase, that means removal of hydrogen. So how we are going to remove hydrogen? NADH, NADH2 will be released as NADH plus H plus. So what happened here? We are going to get hydrogen in this process. Okay. During this process, during this step, we are going to gain NADH2. Okay. Fine. Yes, guys. Okay. So this is NADH, NAD. Okay, NAD gets converted into NADH plus H plus. Okay, fine. So that means here we are going to gain our first payoff energy currency. But you know that this energy currency is not your substrate level phosphorylation. This is an oxidative level phosphorylation. What is oxidative level? That means you need to oxidate it later phases. Okay, that means here we gain oxidative level NADH2. That will be oxidative level. That means each NADH will carry 3 ATP, but you will not get it during right now, and you need some another one step to extract ATP from that NADH. Okay, so that is the first step here. Okay, and one more thing here, everything is going to consider as 2 times. Why it is twice? Because we know glycerol 3 phosphate is an isomer of dihydroxyacetone phosphate, right? So here we consider this also as 2 molecule. Got it? So whatever happens here, everything is considered as twice. Okay, so that means 2 NAD is going to get converted into 2 NADH plus H plus. Got it? Okay, so how many NADH2 we have received? We have received 2 NADH2. Okay, so this is the first step, and remember, this is one of the important step. Okay, and this step is also reversible. This step is here, after all the steps are reversible. Okay, so you say that here in this, like in the first step, we saw that first step and the third step is irreversible. Rest of all the steps are reversible, and if it is irreversible, that means I'll call it as regulatory step. So how many regulatory steps are there in the preparatory phase? 2 regulatory steps are there in the preparatory phase, and let's see how many regulatory phases are going to stay in a payoff phase. I'll directly say it is 1. Okay, so let's see where that 1 is going to be. Okay, so this is 1 for 6th step, where it is not a regulatory step. This is a reversible step, right? And now this 1,3 bipega gets converted into this is considered as 3-phosphoglycerate. So now the question is, again, this is twice. Okay, that molecule changes won't be there. Okay, so this is 3-phosphoglycerate. Okay, so what happens here? Initially, you can see in this case, here you have 2 phosphate. Okay, here you have 2 phosphate. Am I right? Okay, biphosphate. But here we have only 1 phosphate. So what does it mean? That 1 phosphate has been released out. Okay, that 1 phosphate has got released. So ADP gets converted into ATP, right? So that means how many ATP released? We know that everything is twice, so that means it is 2 ATP. Got it? So this is the first substrate level energy which we have received during this process. Got it? This is a substrate level phosphorylation. Okay, so 3-phosphoglycerate is produced. Now next, see, this is again a reversible reaction, and now the next reaction is again is a reversible reaction, which is 2-phosphoglycerate. So here you say that, like, did I remove any phosphate here? No. The position of the phosphate has got changed. Initially, I have phosphor in a third position. Okay, now the position has been changed. It's just an isomerism. Got it? And here we don't do any other steps. Okay, done. And then 2-phosphoglycerate becomes phosphoenol pyruvate. So now this 2-phosphoglycerate gets converted into phosphoenol pyruvate. Okay, again the same two molecules. So what happens here in this case, that glycerate has got converted into pyruvate. Okay, so that means the form is getting differed. Okay, so in that case, how it happened? Some reason should be there, right? That means water molecules has got released during this step, so that the glycerate has got converted into pyruvate. Okay, so we know that how many 2-phosphoglycerates are there? We have 2 molecules of 2-phosphoglycerate. So if it is getting converted into 2 molecules of phosphoenol pyruvate, that means how many water should be released? Again 2 water should be released. Okay, fine. And coming to the last step, yes, this is the last step, right? So this is the 7th step, this is the 8th step and 9th step, and coming to the last step, which is a regulatory step. Okay, if I mention it as a single arrow mark, that means it is a regulatory step. Okay, and remember, write it also, this is a regulatory step. Right? So what happens here? This 2-phosphoglycerate gets converted into phosphoenol pyruvate, get pyruvate. That's it. Okay, phosphoenol pyruvate gets converted into pyruvate. What happens to that phosphor? What happens to the phosphor? Very simple. The phosphor is released in the form of ATP. 2 ADP gets converted into 2 ATP. So here we have received some amount of ATP. So is it a substrate level or it is a oxidative level? Answer is, this is also a substrate level. Right? So finally we got pyruvate, 2 molecules of pyruvate. How many carbon compound is a pyruvate? All are 3 carbon compound. Okay, so this is 3 carbon, this is 3 carbon, this is because we didn't, in nowhere like we released carbon. Okay, nowhere like decarboxylation takes place. So the carbon amount remains same. Okay, so what is the number of carbon in pyruvate? It is 3 carbon compound. And how many molecules we have received? We have received 2 molecules of 3 carbon pyruvate. Okay, we call it as pyruvate or pyruvic acid also. Okay, I hope this is very clear for you, and every single step has a reason, and hope you understand what is that reason also. Okay, and moreover you understand, like sidewise I gave the steps numbers as well, and here I explain whether it is a substrate or oxidative also. And coming to the balance sheet, okay, in the preparatory phase we spent 2 ATP. Okay, we spent 2 ATP in a preparatory phase in step number 1 and in step number 3. Am I right? Now here we have received, we have gained energy. In which all the steps we gained energy? We gained energy in 3 steps. Okay, so what are all that? First one is 6th step, another one is 7th step, and last one is 10th step. Okay, in the 6th step, what we have received? We have received NADH. Okay, and in 7th step ATP, 8th step ATP. Okay, so we know that NADH2 is oxidative level phosphorylation and 7th and 10th is substrate level phosphorylation. Got it? Okay, so again the question is how many NADH2? How many NADH2? We know that here we receive 2 NADH2, similarly 2 ATP and 2 ATP. So total here we have received 2 NADH2 and 4 ATP. Okay, I already lost 2 ATP, right? So what I'm going to do, I'm going to compensate that. Okay, I'm going to compensate that. So I'll get net 2 ATP. Okay, I'll get net 2 ATP. So finally, out of breaking down of one molecule of glucose, by breaking down one molecule of glucose, how much energy is released? 2 NADH2 is released and 2 ATP is released. So remember, ATP is the net. Okay, I didn't calculate that 4 because the earlier preparatory phase we spent 2 ATP, right? From our pocket we took it back to us, and so it is totally 2 ATP. And this is how glycolysis gets completed. And this is if you completely go through this video, I assure you for sure you will not have any blocks in this topic, and wherever the question comes and you will be able to easily attend. Okay, so main point to remember, we have 3 regulatory steps. Okay, which are all the 3 regulatory steps? 1, 3 and 10. So important. So that's it. So with this I'm winding up today's session and we'll meet in another session.